Welcome back, everyone. Chapter 8 is going to be dealing with microbial metabolism, and it's going to be a pretty heavy chapter. If you are feeling uncomfortable with your chemistry knowledge at all, I suggest going back and reviewing some of that. Even chapter 2 might not be enough of a review for you guys with a refresher for that chemistry stuff. So when we start getting into this and you start feeling like you're a little bit, in too deep with regards to that, feel free to take some time, go back, refresh your memory with that chemistry knowledge, because there's going to be quite a lot of it in this chapter, and it's quite heavy, and it carries on throughout the rest of the semester, particularly in the lab. So if you're having a hard time understanding what's going on here, take the time to take some steps back. That's okay. Right? I don't expect you guys to be chemistry majors after, one semester of basic chemistry. So this is difficult stuff. This was difficult for me to learn when I took biochemistry in, my 4-year college. So it's okay. Let me switch this around. Okay, so metabolism is what we're going to be talking about, specifically microbial metabolism, but we will be talking about just metabolism in general, which brings me to a big question that I just want to start off with right off the bat. Of course, this is not like for credit or anything, but I do want you to take a minute to think about it. If you know the answer or not, and I bet you don't, and that's kind of the point here. We all know that you and I breathe in oxygen and exhale, breathe out carbon dioxide. If I were to ask you guys why that is, I imagine that some of you, your answers would be, well, the red cells, we breathe in the air and the red cells pick up the, with the hemoglobin, they pick up the oxygen and take it to the tissues, and then they exchange it for the waste product of carbon dioxide, and we exhale that blah, blah, arteries and veins and all this. That's not what I'm talking about. What I'm talking about is, why do we need the oxygen? We breathe it in, yes, and it gets carried by the red cells to the tissue so that all the cells can have access to it. But what are the cells using it for? And why do we exhale carbon dioxide? Like, where did that come from? Like, are they in the same, like, are they related? Is it the same process going on there? And we're going to get into all that. We're going to answer that for you today. This might blow your mind because it's kind of nuts where how it all happens, but this is how we're getting into all this stuff. So I have to click. I always forget to click. I don't know why I do. Okay, so what we're going to learn about in this chapter, what enzymes are and how they function. Electron carriers used in cells. Boy, we're already getting like into the depth of it, right? Glycolysis and reactions that can occur after that, the Krebs cycle. Wow, okay, we're getting real heavy. Catabolic pathways and the similarities and the difference of the different catabolic pathways. Already, these are all words you don't understand. Where the carbons and the ATPs are going in aerobic respiration, kind of getting a little bit, maybe a little bit familiar words at least here. And then how fermentation works and what it is, and examples of products that we might use as a result of that, right? We're also going to get into how photosynthesis works. Yeah, like I said, this is going to be heavy, so get ready, okay? Metabolism, what is it? We're talking about all of the reactions and workings inside the cell, literally all of the chemical reactions. And remember, everything that happens anywhere is technically a chemical reaction that happened, okay? So even me moving my arm right now, chemical reactions occurred in order for that to happen. My voice like this, chemical reactions happened in order for that to happen, right? So all of the chemical reactions and workings of a cell is talking about the metabolism of that cell, okay? We can divide it into 2 parts. Anabolism, that's biosynthesis. We're making big molecules out of smaller parts, right? So making new things. Catabolism. Catabolism is breaking bigger things down and releasing energy so we can use it for anabolism, right? So they're usually linked together, right? The way I remember is catabolism. Cats are pretty destructive. They come up when they rip apart your couch and all of that. Cats are going to break things down. Catabolism break things down, okay? So in all of metabolism, what are we doing? We're assembling small molecules into large ones, right? And in order to do that, we need energy. In order to bring pieces together to make big whole, we need to put energy into it. And we do that in the form of using up ATP, adenosine triphosphate. Okay, we'll get into it more. So we can degrade large things into smaller molecules in order to get energy to be harnessed in the form of making ATP. Okay, that's our energy molecule. Okay. Another way that energy is lost here, of course, is heat. That is a natural thing that happens in a lot of reactions. So we know that that could be a part of the process as well. But we're trying to conserve as much of it as we can into the form of ATP for use later on. So this is how they're kind of linked all together. We have catabolism here, breaking things down and making energy. So we are yielding energy here. And then we're going to be using that to make things like building blocks like amino acids, or putting those building blocks together to make whole bigger things like proteins. And we can bring all these proteins and, you know, sugars like peptidoglycan, that's made of sugars. Okay. Nucleic acid, they have sugars in them too, by the way. They're made of nucleotides. And then we have fatty acids. Now remember what a nucleotide is. If you don't, you might want to go back and look at it, but it is a sugar and a phosphate linked to a base, right? So those are nucleotides, a nitrogenous base. Anyways, we bring all that together to make a cell. That's what's going on here. That's how we build cells. That's how we repair cells. That's how we, you know, do more things inside of the cell, get things done in the whole organism. Right. Some organisms are just, what are you doing? Sorry, what's she doing? She's on the back. What are you doing, honey? She's on the back of the couch. Ripley, just lay down. Lay down, mama. So anyways, getting into enzymes. Enzymes are just basically biological catalysts. And we call it a catalyst. It's what, you know, it's probably a word that you've heard before. When you're catalyzing something, you're helping it happen, right? You're sparking it. You're giving it the energy that it needs in order for it to occur. You don't have to keep smelling me. Nope. Really? I'm so sorry. So catalysts are going to help get a process done faster, quicker, overcoming the energy. It's going to help overcome the energy that would be needed otherwise. So in order to go from A to B, you might need a certain amount of energy. To go from A to B, you might need a certain amount of energy. It could be, I don't know, which arbitrary, 50 energy, whatever that means, okay? Doesn't matter. But we add in an enzyme to help everything. That helps us overcome that energy. Now we might only need, we might only need like one energy to get this guy to make A go to B. So that's the idea with enzymes. That's what enzymes are going to do. They are catalysts. They are biological catalysts. So they're helping a reaction go along a little bit faster, a more controlled situation going on there, okay? They overcome what we call the activation energy. So that's the energy that has to be put in, that 50 that we're talking about, helping the whole thing overcome the energy that's needed for it to happen. So what is going on? What are the characteristics of enzymes? How do they work? What are they going to do? Most of them are protein. They might have cofactors. We'll get into that soon. And I'm not going to go through all of these, but these are, we're kind of go through some of this here in a little bit. But go look through this, get an idea of what an enzyme really is. It's just a protein that's going to help a chemical reaction occur. We're lowering that activation energy, the energy that's needed to make that reaction occur, with that enzyme changing its shape. Okay, so it's literally going to change its shape. And by doing that, we're helping overcome that energy, because this is all just chemical reactions, right? And the enzyme changing its shape is a chemical reaction too. So instead of using a whole bunch of energy to get from A to B, now we will put a little bit of energy to change that enzyme shape, and then that's going to help A go to B. That's the idea there, okay? They're pretty specific. A lot of enzymes are pretty specific. They act on certain things, and they're usually a lot larger than whatever they're acting on. That's important because they have to, recognize it and act on it, and usually be regulated as well. And we'll get into all that. And they're, of course, they're affected by pH, right? High or low. Why would they be? We've talked about the hydrogen bonds, right? Hydrogen bonds are, positive aspect to hydrogens that are bound onto other molecules because their electrons are being hogged by other things, right? So the hydrogens have a positive, they'll interact with the more negative things. That's a pretty important force as far as making the shape of proteins the shape that they are and how they function. So if you start introducing a whole bunch of hydrogen ions that are free in the solution, remember H+ is hydrogen ions. We also call these protons, okay? we introduce a whole bunch of these into the solution. That's going to affect how the hydrogens and the negative things interact with one another in the molecule, like of the protein and its shape, right? That makes sense because they're interfering. Same thing if we take away H plus or introduce the opposite of that, which is that hydroxyl group, right? These hydroxide ions, essentially, these guys represent basic. So the more of those you have, the less of the H plus that you have. Okay, so you can work either way, but increasing pH, making it more basic, can affect how those enzymes can function and do their thing too, as well as their shape overall. So how are enzymes going to work? How are we going to break this down? Right. So an enzyme is the thing doing the action, right? So if an enzyme comes along and we'll just use the one that's down here, right? And we'll call this is an enzyme. This is the thing that's going to allow us to overcome that energy that was needed, right? So this, in our example from before, we said we were taking A and making it into B. This would be A. Okay. So A would come along and fit into its special site in the enzyme here. And the enzyme will then change its shape. And in doing that, it will allow A to become B. Okay. So it's pretty specific. The enzyme itself isn't becoming part of the product or anything. It is changing, but it can revert right back. So it can function over and over and over again, as long as conditions are asking it to, right? Conditions are favorable for it to do so, as dictated by what's going on in the cell, right? So it's not going to be used up. It can be used over and over as long as we need to use it over and over, right? So how are we going to control? I just said, as long as we need to, and as long as, whatever, but like, what is that? Why would we need to control it? How would we control it? So we get into the concept of enzyme structure. So we're talking about the proteins here and how they're all, folded up and then they interact with whatever they're acting on, like A, right? They're interacting with their A substrate and, changing it into whatever. Well, if it just does that all by itself and it doesn't need anything else to help it along, then that is a simple enzyme. It's just the enzyme acting on its substrate, and that's all. Okay, so this is talking about their structures now. We can also have conjugated enzymes. That means that they need another part to it. There's at least two parts to it. Okay, the whole thing with the enzyme that needs the part plus the part that it's adding on, that all together is called the holo enzyme or hollow enzyme. I always just remember as the same thing, sounds a lot like whole. That's the whole thing all together. That's what's going to function. Okay, has to be coming together in order to do it. So the part that we have, the enzyme, what would have looked like in a simple enzyme, just the enzyme, that portion, the protein portion, that just the enzyme that's going to be doing the action onto the actual substrate, that's called the apoenzyme. Okay, that's the protein portion that's going to be doing the work on the substrate. The thing that helps it, that's added onto it to make it the holoenzyme, the one that is going to be able to do the function, that part that we're adding on is called the cofactor. That makes sense. We're adding it on, like they're working together, collaborating, right? So it's cofactor. That's a big idea term. So this is literally anything that's going to be needed. The cofactor is anything that's needed to add to the apoenzyme. I'm just going to put apo to make it the functional holoenzyme. That's holo, not hoto, okay, version. So that's what's going on there, okay? So that's a big idea, cofactor. There is a type of cofactor called a coenzyme. It is, as we can tell, it's probably going to be a protein or another organic molecule that's going to get involved with that apoenzyme. So it's another organic cofactor. So the cofactor is a general idea of anything that the apoenzyme needs to combine with to work. That could be non-protein, could be non-organic, could be a metallic ion or something like that, right? When we think of hemoglobin, there's a cofactor there. What's that cofactor? You guys know what it is. It's a metallic ion. It's iron. Yeah, same idea here. Okay, that's the concept we're getting into here. So they need other things in order to function. They allow them to do their job. Without them, they can't do their job. If they are an organic molecule, not like iron, which is metallic, but if they're an organic molecule, which we're going to talk about a lot of them, but those are called coenzymes. Okay. So apoenzyme, go down here at the bottom, apoenzyme, the part that's going to be doing the work, combines together with a cofactor, the thing that is going to allow it to do its work, but isn't going to be doing the work itself. Together, that makes the actually functional whole enzyme that will be doing the work. Okay. So we can have little combinations we see down here on the bottom here. We could just need maybe a metallic cofactor, maybe just a coenzyme, an organic thing to stick onto it, or we could need more than one. It could take more than one. It can take several even. It doesn't really matter, but the concept is the same there. So apoenzymes, the things that are going to be doing the work that may need a cofactor, all right? They have something called an active site. That's the same thing as if I were to call a catalytic site. That's the site, the part of the enzyme that is doing the work. It's going to take hold of the substrate and actually do the work on it in this portion of the enzyme, right? It's that chemical area where that reaction is going to be going on. So it says here, oh, my nose is so running right now. Hazel, just lay down. My nose is so runny. Thank goodness. So we have like typically a groove, and that's why we have it drawn down here, right? Like you can see how that's going to match up with this, right? So it's a groove, but it's going to be a chemical groove, right? How the molecules are going to interact with one another, and how the blue guy, the A, as we were talking about earlier, is going to have chemical interactions going on inside of that active site. Is very specific, right? Could be hydrogen bonds, could be positives and negatives, could be hydrophobic aspect of it, could be a whole bunch of stuff altogether. But that specific interaction between the enzyme in that active site and the substrate, which is going to be acting on, in this case, A going into B, turning it into B, that's very specific interaction, right? Okay. So all of the enzymes are different from one another. They all have a very specific primary structure, which is just their amino acid sequence, remember, and how they've been folded. Remember, secondary is those alpha helices and the beta plated sheets, right? So this is where that stuff is coming in. This is a picture of it. This is a model of what a protein would look like, an enzyme would look like. And you can see they have depicted here like alpha helices and beta sheets. There you go. That's what we're talking about. That's exactly what you guys learned about. So now you're getting to put them into play here. So there's different ways that enzymes can interact with substrates, and it's important to understand how the specific interaction is going to occur. Here we have a basic simple enzyme going on here, right? Just holding on to A and turning it into B. So enzyme is going to bind with substrate, and this calling it S here, that's fine. We'll just call it S. So it's going to turn substrate. It can bind specifically with whatever the starting crab is, right? And it won't fit this. This is a different shape. It doesn't fit the chemical makeup there. Okay. And so the enzyme can then interact specifically with that and then allow that chemical reaction to occur, which would have been very energy taxing to make it occur in otherwise without the enzyme. And now we can break it into parts. We've broken up our substrate into some parts. In this case, this is what we're talking about with regards to what's going on in these reactions here. And that would be our products. So that would be like B or whatever, okay? So broken it down. So cofactors and coenzymes. So remember, coenzymes are cofactors that are biological, organic in nature, okay? Carbon, hydrogen makeup. That's what organic means. So we have a whole bunch of metallic cofactors. We just talked about iron. That was a great example, but there's other ones that can have similar effects, and they can help the enzyme interact with the substrate in whatever way, either by changing the shape of the enzyme that it wouldn't be able to do otherwise without that metallic cofactor, or interacting directly maybe in that site. The metallic thing won't be doing the work itself, but it might help with the interaction there. So it could interact in a couple of different ways. And usually a lot of times in that way are going to participate directly in the chemical reaction. So they might not be literally, change permanently in that situation, but, interacting with other molecules will allow that side to be that like metallic cofactor or whatever to be directly involved in what's going on. So usually that's the case. So now we have coenzymes, which are just organic versions that are going to interact with the enzyme and help it to work. They are usually going to have a chemical group removed from one substrate, from one part, and adding onto another substrate, or they can carry and transfer hydrogen atoms from one thing onto another, or electrons from one thing onto another, or carbon dioxide, or amino acids, or whatever, or amino groups and whatever. So they're going to help the enzyme doing all of this stuff, right? So they can work in a way in conjunction with the enzyme to get that stuff done. So what kinds of enzymes are there out there? We have oxidoreductases, and we're going to talk about these things called redox reactions. I'm going to tell you a little bit about what that is soon, but they essentially are going to be moving electrons back and forth, and that can be in the form of hydrogens. And dehydrogenases are a kind of oxidoreductase where we are moving electrons in the form of hydrogens. Okay, so that's one. Transferases, literally putting a group of one thing onto another thing, transferring it. Hydrolases, okay, so this is a little bit something to be aware of. In hydrolases, we are adding into the reaction. We are adding into the reaction water. Water is necessary in that whole situation to break up bonds of other molecules, okay? If we look down here at ligases, these are things that are going to allow for formation of bonds. We'll add in ATP as an energy source to form bonds and then remove water. So it's kind of the opposite of what is going on with hydrolases. So these are breaking bonds, adding water. Ligases are forming bonds and removing water. Okay, so do remember that these are kind of related to one another. Okay, then we have lyases, L-Y-ases. Okay, these are going to be removing groups from double bonded things. Don't worry too much about that. And we have isomerases that can just change proteins from one form into another form, or another. This can be only proteins, molecules from one form into another version of its form, because we can have, like, for example, in amino acids, L and D forms of them, and we can switch them just in their isomers. They're still just, you know, alanine into alanine, but it's L-alanine and D-alanine, like mirror images. Okay, moving on. And exoenzymes. These are pretty straightforward here, okay? Exo, this is going to be stuff that's going on outside of the cell. And then endo is using the enzymes on the inside of the cell. That's really all we need to know. If we're doing metabolic things inside the cell, creating the energy to make new proteins or breaking down proteins and all that stuff, most of that stuff, metabolic pathways, making and breaking, right? That's going to be involved in the endo stuff. So stuff on the outside is going to be breaking stuff down outside of the cell. Usually that could be harmful things, or making like toxins to attack other cells or something like that. Okay. All right. Microbial enzymes in disease. So this is kind of what I was talking about. Anything that helps the pathogen take hold in its host and maybe even create disease, that could be part of them succeeding as a pathogen, right? Is called a virulence factor. A virulence factor is anything that helps the pathogen succeed in the host. A lot of times that causes harmful effects to us, like things like toxins, right? So a toxin is anything that's going to cause a bad effect to us. This is just a term referring to that, but it's something that a pathogen is going to make that causes a negative effect on us. So examples of enzymes that are harmful to us but helpful to the microbe, it might be something like penicillinase. That's the only one I'm going to really get into right now, but penicillinase, what does it sound like it does? First of all, ACE is something that is like an enzyme. Anytime you see that, it's probably going to be an enzyme, right? So penicillinase is going to break down penicillin, okay? So bacteria can produce penicillinase and then become resistant to penicillin drugs. So that's an example there. It's an exoenzyme, something that they make and put outside of the cell. All right, it's important to understand also that enzymes are sensitive, just like any other protein. They can become denatured. And then here I'm talking about specifically bonds that, like those hydrogen bonds, when we add heat into something like that, for example, we're going to start with heat here. It's an easy one to examine because of when we cook chicken, right? When you cook chicken, it doesn't look the same as it did before, right? Once it's been heated up and cooked, because we've denatured the proteins in that chicken breast or whatever, your cooked thigh or whatever it is, that meat. It doesn't look the same because you denatured it. You changed the protein. The protein has been broken down and unfolded a little bit. That's called denaturation. So no longer has its like natural, normal, functional form. Think of that as the natural nature part, okay? It has been made able to do what it was going to do normally, okay? Taken away the natural form and function of the enzyme. We're breaking it down in a way, or unfolding it in a way. We can also do this with like DNA and stuff too, which is important for later on in this course, but we can do that with heat. We can also do that with pH, which we just kind of talked about a little bit, right? If there's too many hydrogen ions in the area, that can interfere with how that protein interacts with itself or with other things, right? And if we have too little of it, same thing, right? Doesn't interact correctly how it's supposed to, as well as other chemicals can cause problems with this as well. So that causes problems with the shape. It can't bind with its substrate as a result, and it can't function at all as a result of that. So a lot of times when we talk about a metabolic pathway, there's a lot of steps involved because we would have to go, we can't just go from like starch, which has a whole bunch of sugars linked up one to another, and then breaking it down into, I don't even know, like making energy out of it into like yielding what we need as ATP. And we wouldn't make it directly into ATP, but there's steps involved to get it there. In order to get to that point, there's a lot of steps. We have to break the sugar parts down. That's starch is a bunch of sugars linked together. We have to break those sugars apart. We have to make them useful in the pathways that we normally would use to make energy and everything like that. A lot of steps in there. And then the pathways, direct pathways themselves, the simple ones, which we're going to talk about here, those are also a lot of steps. Okay, so that's the idea. Each step has a specific enzyme and a specific substrate that act together to make the thing that goes into the next step. And that's how each of these steps work. And that part should make sense to you guys, right? Just each step works that way. And then we can also have some that can, like the result, like if we're making, into this one goes into A and this one makes a B and whatever. And then we can use B to make, I don't know, our energy ATP in however we get to that. But then B can also be made into C that can also be turned back into A somehow. And then feed back in and feed in the loop. You keep adding back into it based on, whatever molecules we can add back into the cycles. That's a cycle. That's a cyclic form of a metabolic pathway, right? So we are going to be talking about different versions of these. Linear, straightforward. A, the enzyme here, it's going to help A go into B. Enzyme here, B is going to C. There's an enzyme here, C into D, and so forth. Pretty straightforward. What we're going to talk about with regards to this is glycolysis. Next, we have the cyclic ones I was just telling you about, one step to each step and eventually can be fed back into itself. An example that we're going to be doing there is the Krebs cycle. Don't worry, you'll know what they are by the end of this. And then we have branched versions where you could have, like we're talking about with protein catabolism, breaking down proteins, or making amino acids from starts, right? So we could be going from different parts and then a different part, and they could be changed in certain ways and be brought together to make like a precursor that can go down different routes or whatever to make different kinds of amino acids. Makes sense? Whereas we have a protein, we have this protein, can break it down into this form, and then that could be broken down via this way with this enzyme number 1. But if this enzyme number 2 is acting, it will give you this product down here. Okay, that's the differences. So that's metabolism. That's not all of the reactions that can happen, of course, but that's the idea of the different ways that they can work. Now, in order to control all this, we need to be able to control all of this. In order to understand how they're controlled, we're going to talk about how they can be inhibited. So something called competitive inhibition is what it sounds like, okay? We have a molecule that looks like the substrate. So we had this guy here that had our little bite, and then this is our substrate that can fit into it, right? Let's say we have another guy that comes along that looks just like that thing. And it can come in here, and now instead of binding with A to make B, now another thing can come in here. We'll just call this question mark because we don't know what it is. It'll come in here and bind in this site so that A can't bind. That's competitive inhibition. There would be binding in the same spot. The enzyme cannot work. It will be shut down as a result. Okay. Competitive. The substrate and the inhibitor, same spot. Non-competitive. We have the substrate going into one spot normally. The inhibitor goes into another spot. When the inhibitor goes into that other spot, the enzyme can no longer interact correctly with the substrate. Okay, so that's two spots that your enzyme, your protein is going to have. We have your binding site or active site, and the regulatory site where the inhibitor will act. Okay, so that's going to be regulated by molecules rather than substrates. Other than the substrates, you know, aside from the substrate, what we're usually going on. So if we have that inhibitor that's going to bind at that regulatory site, we call that site, also we call that the allosteric. Doesn't really matter. I'm not going to bring this up again, but there's that term in case it is a definition. You might need to know. That's the allosteric site. The inhibitor will bind there, and your enzyme cannot do its job on the substrate in its other part in the active site anymore because it changed. So here's an example of these, right? Over here, the bluish purple. This is A, the normal thing that we would have acted on, right? And this is the thing that has a similar shape. This is question mark or whatever. And it comes along, right? So A, normally when it binds here into its active site, will allow us to turn A into products, B or whatever, right? However, competitive inhibitor comes into the same active site, okay? And now it interacts in that enzyme. No product is made because we're not acting on the substrate. It's just mimicking the substrate. So substrate can't bind, can't make the product. We don't get the product as a result, okay? The non-competitive version, we have the substrate that normally binds in its active site, and it would just go from A and then become a B or whatever. Okay, that's the normal steps. However, now we have this regulatory site over here on the side. So when the regulator is present, and I'm going to show you why they look the same in a moment. When the regulator is present, the thing that's going to stop everything, right? It binds in a different site on the side here. And then it changes that shape of the protein, right? Because a chemical reaction has occurred. They're reacting to bonds in the other part of the protein, causes it to fold differently. No longer can we bind with the substrate. And so the substrate can't sit there. It can't be turned into the products. Now, why do they look the same? Why does a product look like the inhibitor? Because sometimes you have chemical pathways in your cells where you might need this product, right? But if you're putting energy in, and components in and using up things that you have in a cell to make that product, but you don't need it made continuously. You only need it made at certain moments. Then the product, once you get a certain amount of it available, will start shutting down the action of this enzyme to make the product because you just don't need any more of it. Okay, so that's how it signals the enzyme to stop making that yellow product. Okay, stop making B. We don't need anymore. We've got enough of it. Stop making it. Okay, that's what's going on there. So that brings us to the idea of constitutive enzyme. This means it's always there. It's always doing its thing no matter what. Not being shut off, okay? We just kind of always need it. And then regulated. So that's what we were just talking about. A lot of times we have production of a product that can be turned on or turned off in response to other things in the chemical steps. Okay, so you can turn it on or turn it off. It can be turned on, meaning induced, or turn off, meaning repressed. Okay, And yeah, it helps you think of like inducing like inducing labor or something like that. Okay. Making it happen. We're turning it on. Turning off, we're repressing it. Okay. Like repressed memories, they're not there. You can't have access to them. That's the idea with these terms. Okay. And that usually has something to do with changes in the concentration of the substrate itself. So the thing that we're binding on, or the concentration of like the products. All of that can come into play with regards to regulating an enzyme. So here is how the big picture works. We have DNA, right? We're turning it into RNA. That's called transcription. We'll get into that more in a later chapter. That's transcription. RNA will be then translated into protein by those things called ribosomes. Remember, I just want you guys to keep that fresh in your minds. Okay, so then we've made our protein here, and then it will fold up appropriately, and then given the right environment, into its functional enzyme form. Then that enzyme plus whatever it is binding with, A, can turn A into B, the products. Okay, we made our products. Those products, when we get enough of them, can actually literally go and interact with the DNA itself in that gene area. And that tells the cell, stop making the enzyme. So it will stop it there. So we won't even get any more of this. So that's how that can work there on that level. So how do we control enzymes? We're talking about controlling whether the actual enzyme could be made itself. So when we talk about making the protein, we're talking about the enzyme itself. We can control the enzyme production itself. That's what we're just showing over here. So we've made a product down here. Product comes, interacts with the DNA and tells the DNA, essentially inhibits it from making the enzyme. We don't need any more. Don't make the enzyme, right? So that's the idea there. We can control the making of the enzyme. We can control the presence or interactions with the coenzymes or other cofactors like the metallic ions. We can control maybe the pH in the environment if we're dealing with a specific, pumping ions in and out of certain areas and whatever like that. That can tell the enzyme to stop doing whatever, as can temperature changes. Okay. If there's high temperatures, it might signal the cell to start doing X thing instead of this thing whenever there's lower temperatures. Could be, maybe that has to do with repairing even. Okay. And then literal actual inhibitors, where we were talking about products working in a feedback loop, coming back and causing everything to stop. Okay. So that's the idea there. So now we're going to get into energy making in the cells. We have these things called exergonic reaction. Exergonic, remember X, we like to think of that as being out, and endergonic, N is going to be like in, okay? Okay, so outgoing reactions. What's happening with the energy there? The energy is being released, it's going out. So we had a bigger molecule. To get energy out, we have to break it down. So that must be catabolism, because remember, that's catabolism because cats tear things up. We're breaking things down. Then we have the endergonic reaction, where we have to add, the energy has gone in, right? Energy is going in. So if energy is going in, we're probably putting things together to make bigger things, using the energy that we made kind of up here to make different molecules, okay? And that, since it's not catabolism, that must be anabolism. Okay. All right. So that's how these things are kind of paired together. You can see how that works because, one's releasing energy, one's using the energy, and they can cycle kind of back and forth based on the resources available in the cell. Okay. Big idea here is manufacturing ATP, that energy molecule, and that is done by adding or removing phosphate groups from it. So ATP, adenosine triphosphate, 3 phosphates, we remove the phosphate group. So it's hydrolysis because we're adding in water, okay, just FYI, to break off the phosphate group and releasing energy. We can do whatever we need to with that energy, okay, endergonic reactions. Now we have ADP, adenosine diphosphate, as a product, and we'll have inorganic phosphates just hanging out there. They've been clipped off. The energy went elsewhere, but now we have these byproducts. And now we can break things apart via catabolism in our exergonic reactions to release energy. So we're releasing energy when we break it up. Where's that energy going? Adding these inorganic phosphates back on to the ADP to make ATP. So you see how it's cycling all together. So here's how it's going to work. We're going to start with glucose. That's our basic molecule that we're going to start with. And we're going to put it down a series of pathways and enzymes, breaking glucose apart and then using the energy from breaking it apart to make ATP in a lot of different reactions. How are we going to do that? We're going to be releasing electrons along the way in the form of hydrogens. So when the hydrogen is just a hydrogen by itself, it has an electron associated with it. But if we have a hydrogen ion like this, it's positive because it don't have an electron. So we can have hydrogen ions and actually free the literal actual electrons on there and use those electrons to power the formation of ATP. And that's what we're going to be talking about here. Okay. So as a result, we're breaking down glucose. You see in glucose how we have all these carbons and all these oxygens and everything in here. As we're breaking it apart into its pieces, we've made carbon dioxide. And that is why you exhale carbon dioxide. We're going to get into that. Don't worry. But the other part of it is in making that energy to make, in taking the energy to make ATP and store it as ATP, I should say, we use these electrons along the way. Well, we can't just have hydrogen ions, the H pluses building up, and we can't just have electrons, just where they go. So we take the hydrogen ions with the electrons and take part of an oxygen molecule to make H2O as a byproduct of that part. Okay. Now this is, of course, going to be a lot more complicated, but that's the idea. That's why you breathe in oxygen and that's why you breathe out. That is literally why. Okay. You learned a thing just there. Okay. So in order to get all of this, you're going to have to understand redox reactions. And I am sorry because it gets confusing, but essentially we have, I remember it, you can call it oil rig, or you can call it Leo. I always learned it, sorry, Leo. Let me just start over with that one. You can call it Leo says GER. That's how I learned it, but oil rig might make more sense, whatever. It doesn't matter. They're saying the same thing. The big things here, we have the R with the G's, right? So that means gain equals reduce. Lose equals oxidize. So that's how we split this up here. L and O, O and L. The E's and the I's aren't as important here. Okay. They don't really matter. They're just helping make words so that you can remember it. Losing is oxidizing, gaining is reducing. What do I mean? We're talking about electrons. If you lose an electron, you have been oxidized. If you gain an electron, you have been reduced. Okay. So we have, don't worry about this bond part right here just yet. Sodium, it's got an electron out here, right? Sodium's got an electron out here. Na is sodium. And then the Cl, this is chlorine. It's just sodium chloride, right? So we talked about our ionic bonds and whatever, right? This is what happens, okay? Sodium has given its electron to chlorine here to make it happier. It's happier with 8 on the outside, and this doesn't want just 1 on the outside. They're more stable like this. Just happier, that the atoms are happier, right? So it got rid of its electron, sodium did, and gave it to chlorine, right? So chlorine gained, so it is reduced, and sodium gave or lost, so it's oxidized. We can even look at it even further and break it down like this, because sodium is going to give its electron to chlorine, and chlorine will become reduced. It is reducing chlorine. Chlorine will be reduced. The thing that reduced it, we would say it is reducing chlorine. it will become oxidized in the end, right? Same thing the other way. Chlorine is stealing away an electron, so it is oxidizing sodium. Sodium has been oxidized, lost an electron. How did it do that? Well, it was oxidized by chlorine, which has now gained an electron and has been reduced. I know. Just go through this a few times, see what's happening to the electrons, what's happening with these terms. If it is still confusing, please let me know and I will break it down into more, but we need to move on. Okay. Here are our electron carriers. So I keep talking about in all these cycles that we're moving electrons around and whatever, but they're just going to fly around. They have to interact with things in order to move around. These are the things we're going to be seeing in our cycles. NAD, don't worry what it stands for. Don't worry what it stands for. FAD and NADP, that's going to be really just in photosynthesis. Okay, so that's the things that are going to be dealing with the electrons. They deal with the electrons in the form of hydrogen. When we have NAD just by itself, and we're adding a hydrogen, which is carrying with it an electron, we get NADH. Okay, so NAD is gaining an electron, therefore is being reduced to, so this is NADH, NAD was reduced to become NADH. Okay, that's going on. So we are having reduction here. Something else had to be oxidized in order to get there. We're going to get to what? So moving on, we have catabolic pathways where we're breaking things down, or cats are destructive. They're breaking things down. The electrons are extracted in the whatever process we're doing. And then we're going through redox reactions, right? So moving electrons from one molecule to another is what's happening here. And then in the very, very end, we have the final electron acceptor. In aerobic, literally think of air when you hear that, okay? Aerobic metabolism, oxygen is the final electron acceptor. And we'll be saying this a lot. Final electron acceptor in aerobic metabolism is oxygen. Air, oxygen, okay? Anaerobic, so here we think of aero as air, right? An is not air, okay? No, air, okay? Not air. Not using air. They're using, not using oxygen, okay? They're using other things to do that instead. That's all that's happening. Not oxygen, whatever else. Doesn't matter at this point. We'll get into what. Okay. So anytime we're transferring electrons, we can use that to make energy. That's what's going on here. That's a big picture. Then we have oxidation and reduction in biology. Moving these electrons actually down these pathways to actually get energy. How we do all that, how we're going to store the energy when we get the electron to the final place and we've got all that energy that we've transferred down that whole system, we're going to store it in the form of ATP. The electrons will allow us to perform phosphorylation. That's literally just adding a phosphate, an inorganic phosphate, just a phosphate by itself, sticking it on. This is ADP. This is what this says over here, by the way. I'm not going to move the thing. Sorry. ADP, adenosine di, 2 phosphates on it, adding on the inorganic phosphate, and that puts energy into the ATP. That's because we have a negative phosphate. So if we had a adenosine diphosphate, right? So this is the adenine and its little sugar part, whatever that it's attached to. It's a 5-carbon sugar that it's attached to. And it's got a negative here, which is a phosphate, and a negative there. So that's ADP, okay? We want to add on another phosphate onto that. Now, why would that mean energy? It would mean energy because this right here, that bond between these phosphates, this is a negative thing next to a negative thing. That's like, you know, the negative part of a magnet with the negative part of a magnet. What does that do? They push apart. So we forced a bond to happen there. They want to push apart. They don't want to be there together. So that when we break this bond between the di and the triphosphate, we are releasing out that, wow, well, that was way more exciting than I meant for it to be. We are releasing out this energy that we can use to power other reactions, okay? That's ATP. So here it is, and we're looking at it overall. We get energy from releasing the 3rd phosphate group. It's just looking at it from the opposite direction. third phosphate group, we can get energy from breaking off the second one as well. It's just not going to be quite as much energy. Okay. That would be AMP, adenosine monophosphate, right? Okay. So adenine, nitrogen base, ribose, 5-carbon sugar, and then the phosphates, either 2 for di or 3 for tri. Okay. The repulsion between the phosphates is what's going to hold the energy. We can break that to release it. Right. So we've got an idea of how ATP works and why. So ATP is our main chemical currency, and we're always going to need it. We need to keep making more and more and more of it, because pretty much a lot, if not almost all of our cell processes are going to be fueled by ATP, right? You have runners that are running a lot. Their mitochondria, which is where we make our ATP, they have just crap loads of mitochondria in their cells and their legs, because they need to make so much energy in the muscle cells there. Muscle cells have a lot of mitochondria. They need to make a lot of energy. They need to make a lot of ATP. We will always need to make and make and make and make and make. That's why you can't stop breathing, literally. You can't hold your breath. You need oxygen because you need that final electron acceptor in this pathway. You will die if you don't get it, because you need the energy to keep your cells going. All right, that's why this works the way that it does. We have something called substrate level phosphorylation. That is exactly kind of what it sounds like, just putting a phosphate from one thing directly onto the ADP to make ATP. Okay, just moving the phosphates around. Then we have what we're really going to be talking about the most is oxidative phosphorylation, and that's using our redox reactions, which is our moving. Just look at that as meaning we're moving electrons around, and this is what electron means. Okay, that stands for electron. I guess it's a better way to word that. So we're moving electrons around. Those are our redox reactions from one molecule to another, and then at the final part of all of that, when we've done that enough to get enough energy, we can get enough energy built up. And I'll show you how we do that. We can allow for oxidative phosphorylation, ADP into ATP. So that's going to power turning ADP into ATP, putting that inorganic phosphate on there. Okay. In plants, they can do, not just plants, but there's bacteria that do this as well. Anything that does photosynthesis can use light to trigger all of this. All right, so the 3 basic possible catabolic pathways that we have going on here. Catabolism, breaking things down. We break things down to make energy. Break to make energy. Okay, there's 3 ways we can do it. Aerobic, which remember is going to use oxygen in the end, or things that are not aerobic. Okay, that includes anaerobic or fermentation. Okay, talk about the differences in those in a little bit, but the main start for all of them is glycolysis. Lysis, here we're talking about breaking apart. You can lyse a cell apart as well. That's what lysis means, breaking something apart. And then here, glyco, we're talking about specifically glucose. Glyco is another term for like sugar. Okay, so here we're talking about specifically glucose. So we're going to start essentially with aerobic respiration, talking about glycolysis. Jeez. Talking about glycolysis here, breaking down glucose. Now you notice we have that in aerobic, anaerobic, and fermentation. All of them have glycolysis there, and it's basically the same thing. We're going to have to put in an NAD with a H. We're going to get out an NAD with a H, a little bit of ATP, right? And then we're going to make carbon dioxide as a byproduct during this. Okay, that's glycolysis. And fermentation, we will just kind of stop there. We can break the products down into different things that can result in like alcohols and acids as a result. That's all fermentation is. Okay. If we have anaerobic, non-oxygen using, okay, we will go from glycolysis into the Krebs cycle, just like we do in aerobic. Where's the difference? we had oxygen as our electron acceptor in the third part, which is the electron transport system. Sorry, I just completely erased it out. This is step 1, glycolysis. Step 2, Krebs cycle. Step 3, the ETS or ETC, electron transport chain. Or you can call it electron transport system. It doesn't matter. But basically, oxygen will be the final thing that takes up the leftover electrons and stuff. But if it isn't using oxygen, we'll use just some whatever else we can based on whatever bacteria we're talking about. Okay, so that's what's going on there. So moving on, we just talked about this. It's just breaking it down into word format so that you guys can know. At each step, we could be making NADH, reducing NAD into NADH. That there, that H, that is what I'm talking about with regards to electrons. That H can come off later into that electron transport system down here. And as an electron get pushed through that system. And that's the electron acceptor that we need for, okay? So those electrons up from here. What's going on? Okay. Same idea with all that. And we kind of talked about fermentation a little bit. As far as it's mostly just glycolysis, and then the products from it we can change up to get alcohol and acids as a byproduct from that, right? So aerobic respiration, this is what our cells do. A lot of bacteria do this as well, as do all of the eukaryotes do this. And bacteria can do this. So we have enzyme catalyzed reactions. We have quite a few of them that we're going to be breaking down glucose, making having things, breaking off electrons, putting them onto carriers. Let's just put it that way. We're going to take glucose. We're going to take the electrons from glucose and put them onto carriers like NAD and making it NADH. And then we're going to use these electrons in the electron transport system or chain. Okay, we move it through there. That's going to allow us to create an energy gradient. We'll see why in a moment. And then use that to make ATP in the end. We can make quite a lot of it. All right, glycolysis is the first thing we're breaking down. Glucose, main thing you need to know about this, glucose is going to be converted into pyruvate or pyruvic acid, same word, okay, pyruvate. That's our kind of end goal here. We will make a little bit of that electron carrier NADH. We might make a little bit of ATP as well with each glucose molecule, but the goal here is to set up that pyruvate for the next step. So here's what it looks like. Oh my gosh. Getting into the first part of it, starting off with glucose, it is a 6-carbon sugar. And then we're immediately going to use ATP to add a phosphate onto that. We're going to change it up a little bit, change its shape a little bit with another enzyme, add another friggin' phosphate on. And then we can break that down into these guys down here, glyceraldehyde 3-phosphate. Essentially, we also get this junk, but it's going to get just changed essentially back into the glyceraldehyde 3-phosphate. So we'll just worry about that. Okay. Now we got our glyceraldehyde 3-phosphate. Now what do? We have 2 molecules of glyceraldehyde 3-phosphate, or its sister, the DHAP. Okay. Same difference. So they can each go down. We'll have that glyceraldehyde 3-phosphate. We will lose a hydrogen and basically an electron to make NADH. And then we can add in a phosphate with this. Here we've got another phosphate stuck on. Now we can remove a phosphate to make ATP here. Okay, so this was inorganic. And now we've got this CCC with the phosphate on it. We can remove a water here. That changed it chemically to another thing. Don't worry what they are, but here is what they are. If you're, for whatever reason, just, a sadist and want to know what's going on there chemically, there's the words for them. We've changed their shapes, essentially. We've removed finally that phosphate. We've gotten to this point where we can remove the phosphate, and now we can make pyruvic acid or pyruvate. This is the goal. Why? We've made a little bit of ATP. We made an ATP here and an ATP here. And you will say, wait. You just told me in this first step that we used like 2 ATPs. Yes, we did. But because we get 2 molecules here going into the second part of this, then we are able to, so this is the second part, sorry, breaking it down again. So we get these 2 molecules, all you have to worry about. Each one gets to go down to make 2 ATP. So there's 2 ATP made over here, and we've made up, now add a neutral here, and we made 2 over here. Okay. We made an NADH here, and we made an NADH here. So we've made 2 NADH there. And we can use those in the electron transport chain. So I can't remember where one more. So that's what we have now in the end. Right. We've set it up with pyruvic acid or pyruvate. It's very important. We can get here from pyruvic acid. We can get here from other things like amino acids and stuff. We can also use pyruvic acid to make things like amino acids or other larger particles or whatever. This is also what things in fermentation, if you're fermenting, here you would take pyruvic acid and turn it into acids or acid aldehyde, which would turn into alcohols. Okay, so that's kind of what will be going on there. All right, so moving on to the Krebs cycle. The Krebs cycle, we have now taken that very valuable pyruvic acid, it's important intermediate. We'll turn it into this thing called acetyl-CoA and go into the Krebs cycle. We are going to start releasing carbon dioxide here. So that's what's going to start happening here. And then we're going to make more NADH. And we'll have a little bit more of that to carry into the electron transport chain. So all of this, the Krebs cycle for each glucose molecule will go twice. All right. So here's the Krebs cycle. You don't need to know all of the individual steps. But basically what I'm going to say here is we have our acetyl-CoA, and at each step where we have changes going on, it sets us up to like make a NADH or something, right? Or we've made carbon dioxide as a byproduct, and then we can do it again, another NADH, make carbon dioxide as a byproduct. And then whatever, we can make a little bit of ATP down here. We can make FADH2, another thing that carries electrons, okay? And then even more NADH. This is important because then we can kind of cycle back in based on what nutrients and molecules the cell has available. It's important because we need all of these electron carriers for the electron transport chain. And remember, we are doing this cycle twice per glucose molecule. Okay, so this is kind of what we've gotten from that. We have made our NADH and FADH2. They are now carrying our electrons that we can use on the next part, okay? We have our ATPs. When this is what I was talking about with substrate level phosphorylation, moving a phosphate from one thing onto another. This is not oxidative on that part. We're about to do that though. Okay, that was substrate level. Now we're going to get into the electron transport chain or system, the ETS, and that is going to lead to eventually oxidative phosphorylation, using all those electrons to make energy gradient to allow phosphate to be put onto ADP to make ATP, and it'll let us make a lot. Okay, so these are our redox reactions. These are our reduced NADH and FADH2. Remember, carrying the electrons. The electrons then pass from off of these guys in the form like the hydrogens, right? So the hydrogen's going to come off. We'll be left with the NAD. Hydrogen's going to come off. It had an electron associated, but now we're putting the electron through the chain. So electron is freed from this. So this is left positive. So this is kind of what's going to be the result of it as we go through the step, okay? So now we're moving the electrons through. Electrons are moving down from molecule to molecule to molecule to molecule. That allows transport of hydrogens. When I say hydrogens, I'm talking about hydrogen ions, these guys. So we'll move them onto the other side of the membrane. Very important step here. And then oxygen at the very, very end, when those hydrogens come back inside, those H plus and the electrons and the oxygens are going to eventually come together to make H2Os as a byproduct. Okay. So this is how it's going to work. NAD gets rid of its little H and its electron. We have an electron that's put in to these molecules here. These are called cytochromes. And yes, you need to be aware of that term. Okay. Cytochromes. As the electron moves from cytochrome to cytochrome, getting them excited, you can think of it that way. Excito. It's not spelled that way, but you at least know now what they're doing. They're getting excited. Okay. This is a wide. All right. So they're cytochromes. They're becoming excitochromes, getting excited by these electrons. As that happens, protons, the H pluses, where we took those electrons from, they're getting pushed on outside of the cell. This is in the cytoplasm. This is inside. This is the cell wall. This is what is called the periplasmic space, or just call it the outside. Okay. But specifically the periplasmic space in bacteria. All right. So now we have pushed all these protons outside. So now we're getting a whole bunch of them on the outside. Then they can get pushed back in because we're creating a gradient. And I'll show you what I mean, through the ATP synthase, which is another enzyme to make ATP. How is this all going to work? Okay. All of this, by the way, is going to happen in our mitochondria. This is happening in your mitochondria right now. The energy this will be going on as far as like the space of what we're creating with the cristae, the folds of the mitochondria. Okay. The hydrogen ions will get pumped onto the outside of that. Within the mitochondrion, though, in that intermembrane space, the cristae fold between the outside membrane of mitochondria, these, in that space is where the hydrogen ions will collect, and they will come back in to the inside area through the ATP synthase. So that's what's going on in our cells. Those are our mitochondria. Okay, so ATP synthase is at the very end of all of this, basically, the electron transport system. And so it will capture that energy that is released by those electron carriers donating the electrons in. Okay, so that's what that energy is going to go. It's going to go into the ATP synthase and allow us to perform oxidative phosphorylation. Okay, so ATP synthase is going to be coupled with that electron movement. The NADH gives its electrons and can result overall, one NADH that goes in can give us 3 ATP. Okay. And FAD, because of how it works, it only gives us 2, just chemically speaking, how it operates. Okay. So that's going to be good to know. So we're going to add all the numbers up at the end. So now we have something called chemiosmosis. When we are moving our electrons, we had all of those cytochromes and turning them into excitochromes, right? We put our electron in, and now they're becoming excited as they move on down. As we are moving that down, we are pumping the hydrogens out. That is chemiosmosis. Okay, that's chemiosmosis. That is going to set up a gradient. A lot of protons are going to come out here. These, remember, hydrogen ions, these H pluses are also called protons. And that's going to make a gradient of them. There's going to be a lot more on the outside part than there is on the inside. Okay, so that is what we're calling the proton motive force. Pushing all those protons outside and using them later on. We're creating the proton motive force. Okay, how we're going to use that to make ATP? created that gradient. Essentially, we've got a lot of positive on the outside of the cell, right? On the outside of the membrane. And we have negative on the inside because we've pushed all that hydrogen outside. It wants to move the positive back in to equal it all out. So essentially, the only way the H pluses, the protons, can come back in is through ATP synthase, which that movement of those positive ions through that protein ATP synthase is going to create chemical or shape changes in that protein ATP synthase. The positives are going to change it a little bit, right? As it moves through. As that happens, that is going to allow for us to start to prepare for ATP synthesis, making ATP, this button, I'm so sorry, out of ADP. So here's how it's going to set up. It's like a dam. We have essentially all of these hydrogen ions that are building up on the periplasmic space or on the other side of the membrane, and it's all like positive over here. So we have the need to want to get out, but we're blocking it. But if we open the floodgates, right, if they want to start coming in, they can come in, but they move through this turbine, and they're going to start turning the turbine, and in turn, we can use that generator to make energy to power the city, right? Same idea as here as with the ATP, like a dam, hydroelectric dam. Whew. Okay, so ATP synthesis in eukaryotes will be the same process, but we'll make it just a touch less because of how things work on the mitochondrial membrane. Okay. All right, so this is the getting to that last step. We've moved our electrons down through here. We've made all these protons out here so that they can come through that turbine, that ATP synthase, so that we can start making ATP. And now we've got a bunch of, hydrogen ions, and we need to get rid of the hydrogen ions. We need to get rid of the electrons, right? And so we have this thing called cytochrome C oxidase at the very, very end. Important enzyme to know. We will be talking about this again, not just in this chapter, but later on in the course, okay? Cytochrome C oxidase will receive those last electrons right here, as well as the hydrogen ions, those protons, and they will use oxygen that we breathe in to make our byproduct of water. So that is what is going on there. Cytochrome C oxidase is what takes care of that. Okay. ATP synthase is what's making the ATP. The thing that's going to deal with the leftovers and why you need to breathe in oxygen, cytochrome C oxidase. All right. So overall yield of how many ATP you can make. This is going to be 38 for prokaryotes. This is not supposed to be there. Prokaryotes, and this is for mitochondria in eukaryotes. Okay, so that's what we're going to yield from all of this, from the one glucose molecule. We can get up to 38 ATP. In glycolysis, we get 2 ATPs. In Krebs cycle, we made 2 ATPs from 1 pyruvate. So we'll get that twice with each glucose. So 4 ATPs. And then we'll also have byproducts of carbon dioxide, and we'll have, making water as a product and all of this, or we're using up water in certain parts and getting rid of carbon dioxide in the form of breaking off, carbons and all of that. So that's where all that's coming from. But yeah, overall we can get 38 tops from aerobic respiration using oxygen. Gosh, keep hitting that button. In aerobic respiration using oxygen, we can get a maximum of 38 ATP for prokaryotes. Eukaryotes is 36. So this is how it's all laid out and where everything goes. We get 4 ATPs from glycolysis and 2 from the Krebs and 34 from the electron transport. The 2 NADH that we got from glycolysis gives us 6. The 8 NADH from the Krebs cycle will be, as well as the ATP from there, we'll have 24 ATP. and then 2 FADH will lead to 4. And so that we can add that all up to 38. Okay, so that's where that's all coming from. This is for eukaryotes. We miss a couple of them because of transporting NADH across the mitochondrial membrane. We lose some energy, potential energy there, okay? What else do we make besides ATP? Well, we have 6 carbon dioxides. We will use 6 oxygen. We will produce 6 H2O, and what we'll use 2 in the Krebs cycle. So, oh yeah, and we produced 2 in glycolysis, right? So we use that, so it ends up being 6 overall. But yeah, that's how that's working out. So pretty equal numbers there as far as carbon dioxide production, oxygen usage, and water byproduct. So the cells can use water for other things, but that's also part of why you need to get rid of water. You need to pee. So anaerobic. So this is no oxygen, and being no, and arrow is O2 oxygen. So no oxygen in these. We can use nitrates and reducing them to nitrites. E. coli does that. We can also turn that nitrogen down into other things. Okay, so that would be called denitrification, taking that nitrate into nitrite and then using it up even further to either nitric acid or nitrous oxide or even nitrogen gas, like what is in our atmosphere, N2. Okay, so this is very important in the biosphere as far as where our nitrogens come from, like in the soil and everything like that. Nitrogen usage, nitrogen reduction, and denitrification are very important for that. But this is anaerobic. This is doing this instead of using oxygen. Okay, so this is fermentation. Essentially, we're just going through glycolysis and using the products of that to get slow production of ATP, and then turning the leftovers into like ethyl alcohol, which is drinking alcohol, or like lactic acid. Okay. Alcohol fermentation, we'll have that happening in yeast and bacteria. We're converting that pyruvate into ethanol or acids. Into ethanol. Sometimes you can have acids as well along with this. Okay, it doesn't have to just be one or the other. So we are now the stepwise thing that's going on. We get that pyruvate from glycolysis, turn it into acetaldehyde, then turning that into ethanol, which is drinking alcohol. So that's what's going on with bacteria and yeast that are fermenting things like beer and everything like that. Okay, so that's what's going on there. And then we can also make acid as a byproduct and not just alcohol, right? So we can have lactic acid formation, giving things like that sour acidic taste. So if we're only making lactic acid, that's homolactic fermentation. Bacteria will do pyruvate into lactic acid only. Heterolactic will have glucose fermented into lactic or acetic or carbon dioxide products. Okay. Bunch of products, not just lactic. Okay. Then we have mixed acid fermentation. There's bacteria that convert that pyruvate product that we get at the end of glycolysis. They're not going to be using necessarily for other things. They can turn it into acetic, lactic, succinic, formic acids, and they can also make CO2. And this is kind of what with other acids that they're making, like butyric and propionic, and then making CO2, we get the CO2 gets the holes in the Swiss cheese, and the butyric acid and propionic acid are going to help with curdling that cheese and everything like that. Okay. These are just some examples of some of the things in our lives that are from fermented products. We make vinegar this way. That's where we get vinegar from, acetic acids from that. We have sauerkraut, and kimchi, of course. Kefir, which is like a fermented yogurt type of a thing. And whatever, you guys know how this stuff works. Yeah. Lipid catabolism. What am I talking about? We're breaking down lipids. Why we want to do that? You're not just going to make energy out of only glucose, right? Our body has to be able to use other molecules to make energy sometimes too. If we're doing this via lipids and breaking them down and putting them into this whole system where we went through glycolysis, we went through Krebs, and we went through the electron transport system, we can enter in at different points with byproducts of other things. Okay, so lipases are going to break down fatty acids, lipids, and then allow them to enter into these cycles at certain points, because we can undergo beta oxidation of the fatty acids that are associated with our lipid molecules, okay? And then use that to create acetyl-CoA to enter the Krebs cycle, so we can get ATP. So if we have a 6 carbon, just 6 carbons, and fatty acids can have quite a lot. Each little dip here is a carbon, okay? Each 6 carbon fatty acid yields 50 ATP, whereas, glucose gave us 38, one sugar molecule, okay? Pretty neat. All right, proteases, same idea here. We're breaking down proteins, protein catabolism here, so that we can get energy from that, and we can enter in this pathway with products related to breaking down proteins, usually like amino acids. And we can have amino groups, which is that NH2 group, that amine group, right? We'll take that off via deamination, and then the carbon compounds that are left over with can be entered into the Krebs cycle at certain points, so we can get energy. So really what's going on in the cell, we have this thing called amphibolism. So we have catabolic and we have anabolic going on at the same place. And we have a lot of intermediates from a lot of these steps, either breaking down or putting together or whatever, that can enter in through a lot of other sets of cycles that are going on in all of the metabolism going on in the cell, right? So the catabolic of breaking things down, very closely integrated with the anabolic of building things up. That improves efficiency of the cell, so we're not wasting too many of these byproducts, right? We're trying to keep everything tight, we're trying to keep everything nice so that we can get as much as we can out of everything. So we have our glucose molecule here. We have glucose that we can break it down, essentially what we were just talking about, going into the Krebs cycle and going into the electron transport chain and everything to make energy. Or we can make byproducts of this whole mess and push them into making fatty acids. We can push them into making amino acids. We can push them into making nucleotides and whatever. But we can also use leftover nucleotides. Like if I eat a meat is going to have nucleotides in it, nucleic acids, right? Those are cells from an animal. So it's got DNA in there. It's got amino acids in there. It's got proteins. It's got starch, or not starch, I guess, in meat, but you know what I mean? It'll have carbohydrates. It'll have fatty acids in there. So my body might want to use that for energy, or it might want to use it for building blocks for other things. If it wants to use it for building blocks for other things, it still needs energy to do that. However, it decides that it needs to use that, it'll work its way out, chemically speaking, right? So that's what we're talking about here. Breaking things up when you need them, putting things together when you need them, and finding where they all fit within one another. Moving on, to photosynthesis. This is using light. Jesus, sorry, this is gonna be a long one, guys. This is using light to make energy. Essentially what we're going to do in our light dependent reactions, the photons will excite the pigments, the color pigments. It's really what we're talking about here. And when that happens, we can start making energy from that, okay? We can power that to do what we want. And then we can go into dark reactions to go on to actually making our energy making molecules, okay? So chlorophyll is the green. It's a light harvesting pigment. It's one of the pigments that we can investigate here. There's a bunch that exist, and this is just talking about down here. You can see a bunch of the other ones that'll be triggered by certain wavelengths of light or colors, right? So photophosphorylation is using those excited photons. Instead of moving electrons down a system, now we're using photons and moving, I don't know what charge, they don't have a charge, I don't think, but we're going to be moving photons down the system and exciting the pigments and everything, right? That's going to pump hydrogen ions and create that gradient there. So we will have NADPH, and we'll also make ATP as a result of all of this. And then that we're going to be using to drive the Calvin cycle, which we call the dark reactions or light independent reactions, okay? So here we're going to have carbon dioxide entering in. This is why photosynthetic organisms need carbon dioxide and use it. So whatever, as far as where this is going, either in the cytoplasm of like bacteria or the stroma in a chloroplast. But this is man, I mean, when you look at it, right? The end product is to synthesize glucose. That's what you need to know. We put light in, we stored it in electron carriers and ATP, and we're using that to make glucose. Why do we want to do that? To go through literally the same cycles that we did in our mitochondria. Plants and other photosynthetic organisms, they have mitochondria too. They're just not getting their energy from food, like glucose from eating a thing. They're literally using the light to make the glucose that they're going to use in their mitochondria. And that's what's going on with their energy system. And that's why mitochondria and chloroplasts are so closely linked together, not just within the organism that has these things like photosynthetic eukaryotes like plants or whatever, or like if we're talking about between different kinds of bacteria, right? It's not just within one organism, but also like outside as well. You know, the oxygen that the plants are producing and our carbon dioxide that we're producing, all of that, right? How that is all linked together. So we can also have photosynthesis where they don't make oxygen. When that happens, they don't make a whole lot of ATP from it, but they will use either hydrogen sulfide, or elemental sulfur as an electron source there. Okay, so not using oxygen there. So these are the strict anaerobes. The strict anaerobes that use light in order to power all these systems. That's the ones we're talking about here. Okay, no oxygen used. They have to have some light added in order to create any energy, because it does not make very much ATP at all. Okay, so this is the end. We made it. Overall, metabolism is going to be coupled processes between catabolism, remember tearing things apart like cats, and anabolism, which is like creating and building back up. At enzymes, now you know what these things are. Enzymes catalyze, so they help convert a substrate to a product by helping over that activation energy, the energy that they would normally need. The enzymes help reducing that overall, okay? They're facilitating the reaction that way. Then we have respiration, which we talked about, which is either aerobic or anaerobic, right? Creating ATP, either using oxygen or not. We talked about the electron transport chain and chemiosmosis, pushing the hydrogen ions, the hydrogen protons across outside so that they can come back inside through the ATP synthase to make ATP, to power ATP. And then we talk about fermentation a little bit, producing lactic acid and alcohol, and then photosynthesis, kind of the opposite of aerobic respiration, as far as they're going to use CO2 and make O2 in the end. So that's the idea there. I hope that you guys understood at least part of that, and I know it's a lot. And then I will see you guys next time in chapter 9, I guess, for more fun.